So have you ever wondered what causes some people to be taller and larger than others? Maybe you've wondered it's due to the genes that are inherited from our parents. However, according to an article called Gut Bacteria That Prevent Growth Impairments Transmitted by Microbiota from Malnourished Children by Bolton, it discusses an experiment that found that microbiota in the gut of humans can severely stunt or aid the development as children, regardless of age or diet. You may now be wondering, what is microbiota? Well, according to the Stanford research, the gut microbiota is a control center for multiple aspects of our biology, including our immune status, metabolism, and neurobiology. In this video, we will discuss the experimental procedures used to investigate microbiota, explain the results of the experiment, and finish off with a discussion.
And now to the experiment. The story of this experiment begins in the African country of Malawi, where malnutrition is a serious issue. In order to find a way to help the people of Malawi with this problem, a group of scientists developed this experiment. They started by meeting 19 children between 6 and 18 months old, some of which were healthy and some of which were malnourished. To extract the gut microbiota, fecal samples were obtained from these children and taken back to the lab. Once isolated, the microbiota were fed to 5-week-old mice, which were provided with a similar diet to the Malawians. Throughout a 5-week period, 3 groups of mice were observed. A first group with microbiota from healthy donors, a second group with microbiota from unhealthy donors, and a third group with half of each microbiota sample. These groups of mice were used to test a couple different experiments.
Experiment 1 tested the effects of the different microbiota groups on the growth of the mice over time. Experiment 2 tested the transmittance of microbiota between the healthy group of mice and the unhealthy group of mice by keeping some mice from both of these groups together. This experiment was termed co-housing. Experiment 3 tested whether certain microbiota founded in the co-housing experiment could reverse the effects of unhealthy microbiota alone. These different experiments produced some very interesting information for the scientists to work with.
Over the course of the first experiment, it was obvious to researchers that mice which had been given microbiota from the children with normal, healthy growth had developed much more than their stunted microbiota counterparts. In fact, there was a 10% gain in body weight and lean mass between the two groups. While weight gain may be avoided by the average person, it's a sign of healthy growth in young children. It was also found that some different species of microbiota were found in different age mice and children, meaning that microbiota maturity is most likely an indicator for host health. These two graphs show lean mass and body mass growth over the course of the first experiment. As you can see, mice with microbiota from healthy children grew up to 10% more. In the second experiment, which tested microbiota transmittance via housing mice together, it was discovered that the co-housed healthy and unhealthy mice had gained more lean mass than the control group of only unhealthy mice, within statistical significance. In addition, the 5 microbiota shown in this table were found to have direct effects on the growth of the mice. Also shown is the lean mass gain of the mice after day 15. Note that the healthy and unhealthy co-housed mice had more growth than even the healthy control. In the third and final experiment, where these 5 microbiota were put into unhealthy mice to test if they could reverse the effects of those bad microbiota, only 2 survived to colonize. They were R. navis and C. symposium. Mice with these 2 microbiota showed increases in lean mass akin to mice with only healthy microbiota. Shown here are the lean mass gain and body mass gain results in this third experiment. Mice treated with these 2 microbiota showed significant increases in both. Devin will now discuss the implication of these results.
So what these results mean for humans is that nutrition is of the utmost importance for growing children. Gut microbiota rely on nutrients gained from the food eaten by the person to provide lean mass growth in return. Healthy microbiota in the gut and the host have a symbiotic relationship because the microbiota can digest organic molecules for themselves as well as for the host. A healthy, well-nourished host has properly digested food that can be used for cellular processes. Undernourishment corresponds to underdeveloped gut microbiota and less lean mass gain in children. This is because cells inside the body require organic molecules for cellular functions. Without these molecules, there is a lack of ATP, which is needed for many cellular functions that are related to growth and development. As mentioned before, proper nutrition as well as an ample food supply are critical for lean mass growth in young children. According to Blanton, therapeutic strategies need to be implemented in order to prevent the stunning of neural development and growth abnormalities associated with malnutrition. However, healthy microbiota can be transferred from healthy hosts to undernourished children, which can help prevent underdevelopment in less fortunate countries. Thank you for joining us today. We hope that you enjoyed.